The three main types of protein receptors, by Natalie Cross, Skyla Sternberg, and Stephen DeCesar. G-protein coupled receptors are the first protein receptors we'll be going over. In G-protein coupled receptors, a signal molecule, aka a ligand, attaches to a G-protein coupled receptor. This causes the receptor to change shape and lets a G protein attach. When the G protein attaches to the receptor, its GDP molecule is replaced with a GTP molecule. This activates the G protein and causes it to move towards an inactive enzyme. The enzyme is activated by the presence of the G protein and changes shape. The enzyme being activated causes the beginning of transduction. When enough proteins are made, the G protein can turn itself off by hydrolyzing the GTP and replacing it with GDP. The enzyme and protein are now inactive and the cycle can start again when necessary. G proteins are extremely important as they are essential for embryonic development and affect other senses such as sight. Unfortunately, though, due to their importance, bacteria can interfere with the function of the G protein coupled receptors and cause major issues. Here, a cholera bacteria is shown disrupting the shape of the G protein coupled receptor, not allowing the ligand to attach. Bacteria like cholera and botulism can prevent the proper functioning of the cell signal transduction pathway.
The next type of protein that we're going over is the receptor tyrosine kinases. Receptor tyrosine kinase proteins come in pairs. Each kinase is an individual monomer. When a ligand bonds to each receptor site on the kinases, the kinase change shape and bind together to form a completed dimer. Though a dimer is formed, the complex is not fully activated yet. ATP must come and phosphorylate each tyrosine molecule for the complex to be fully activated. Once all of the tyrosines are phosphorylated, they change their shape. The change in shape allows inactive relay proteins to bind to specific tyrosines. The attached relay molecules are then activated and begin the transduction process that eventually results in a cellular response. Tyrosine kinase proteins are extremely important and unique because they can activate many different transduction pathways at once, with relay proteins able to attach to each tyrosine.
The next type of protein we'll be talking about is an ion channel receptor. A ligand-gated ion channel is the most simple of the three types of receptors. As the name implies, the channel acts as a gate and is opened or closed as it is activated or deactivated. Gated ion channels are extremely important as they allow ions in or out of the cell. This determines a cell's charge across its membrane, as more or less ions of a specific charge give a cell more positive or negative charge. A ligand-gated channel is closed until a signaling molecule attaches to the receptor. This causes the usually closed gate to open. Ions are on one side of the membrane, then diffuse across the membrane as long as the gate is open and the signaling molecule is attached to the receptor. When the signal molecule detaches from the ion channel, the gate closes and ions can no longer diffuse through it. Ion channels are very important for the functioning of the nervous system, as neurotransmitter molecules can cause these channels to open or close.